Hi everyone. Today I come with the continuation part of the flowering genes. So this is the second part of the flowering genes. You just check out the first part and then come to see that. Okay. So the four major pathways I have discussed in that first part: the photoperiodic, autonomous, vernalization, and gibberellic. The four pathways through which this flowering process is occurred. Okay. So the four major pathways that converge on the meristem identity genes. The meristem identity genes, they are the two important genes, that is LFY, leafy, and AP1, apitala 1. These two important genes that are involved in the flowering process, and that is seen, or its involvement is seen in that four major pathways. The floral integrator genes such as SOC1, FT, and other integrate signals from different converging pathways. So another important gene, that is the SOC1 and the FT gene, and another integrate signals that are also involved in this process, in these pathways where the flowering process is induced. Okay. Under appropriate conditions, the activities lead to increase the LFY and AP1 synthesis. So some conditions or favorable condition, in some favorable conditions, the activities lead to the increased LFY and AP1 synthesis. So in some favorable conditions, this synthesis, the LFY and AP1 gene synthesis process, that become increased. Okay, and that mediate the transition from shoot vegetative meristem to the floral meristem. So simple way is that the activity when increased, the activity of the LFY and the AP1, these genes expression when increased, that help to convert the shoot vegetative meristem tissue to the floral meristem tissue. Clear? Because the shoot meristematic tissue, until converted to the floral meristematic tissue, the flowering process not occurring. Okay. The Constance CO gene, that plays also an important role in this photoperiodic pathway. This is the first pathway. This is the photoperiodic pathway where the light is important factor. And light and internal clock. Internal clock means you can say the circadian clock, and this circadian clock is what I will discuss in another video. Just simple way is that circadian clock is what? That is the day and night that occur rhythmically in a circle. Okay, so this is called the internal clock. Clear? The two important factors, light and internal clock, that precisely regulate the accumulation of the product of the CO gene. So only the CO proteins that is produced from the CO gene, that is, it's not, or that not induce, or that not create the flowering process. Why? Because the two factors here involved. Clear? So three factors needed here. One is the light, second one is the internal clock, and third one is the product of the CO gene. Only the CO gene products cannot, if it is involved in the flowering process, so it is not creating the flowering process to continue, because the two factors here also involved. Clear? The CO proteins directly activates and transcription of the FT gene in the floral meristem. So when the CO proteins produced, this activates the transcription factor of the FT gene. Now CO gene's expression activates the transcription or the expression of the FT gene. And this FT protein now produced, and this FT protein enters the nucleus and forms a complex with the AFD protein. And AFD protein is another protein, and that is formed from the AFD gene. So here CO activates and then protein produced that induce the activity of the FT gene. Then FT protein produced, then FT protein activates the FD gene and the FD protein now produced. Okay, then a complex is formed with this FT protein and the FD protein. After being activated by the FT protein, the FD triggers the expression of the SOC1. This is another gene. When this complex of FT protein and the FD protein is totally formed, then the expression of the SOC1 and the AP1, this is seen. And that encode the transcription factors. Then transcription process will occur, and then both these activate the leafy gene, and the activation of these total genes, the SOC1, AP1, LFY gene, all these are involved in the flowering process. Okay, so the induction of these genes that help in the flowering process of the plant. Clear? Next one is the autonomous and the vernalization process. Okay, here important thing is that vernalization process I have explained in my channel, so you just check out. This autonomous process and vernalization pathway, here the important role is played, that is another gene, that is the FLC gene. This FLC gene's role is what? Its expression actually that stop the flowering process. So there are several loci or the locus. You can say in the singular way this is called the locus, but in the plural way this is called the loci. So there are several loci that, or you can say the regions, that promote the flowering process. But these total regions are repressed by this FLC gene. So flowering will not be occurred. Clear? This autonomous pathway acts by reducing the expression of FLC gene, which is the inhibitor of the SOC1 expression, and in vernalization process that also represses the FLC gene by epigenetic mechanism. Clear? So anyhow the expression of FLC gene must be stopped because this is the inhibitor of the expression of the SOC1 gene. Clear? And until the expression of SOC1 gene is seen, the flowering process also you cannot see. Clear? So the expression of SOC1 gene is very important by directly or indirectly. Clear? So now this expression is completely occurred, and then the flowering process will be continued. And vernalization represses FLC expression is promoted by the, okay, here important gene is that FRI or FRIGIDA, Frigida. This Frigida gene is very important because its activation totally expressed the FLC gene's expression. Clear? So if Frigida gene is activated, that means the FLC gene's expression is also activated. So first of all, this FRI gene's activation must be stopped, then this FLC expression is stopped. Clear? And in case of gibberellin pathway, here the gibberellin plays important role for it promotes the flowering process by activating the expression of the leafy gene. Okay, so here gibberellin plays important role directly. It activates this leafy gene, and in that way this flowering process will be continued. So these are the 4 pathways, and this is the very rough diagram how this process of flowering it is done. So you can see that 4 pathways. One is the autonomous pathway, second is the photoperiodic pathway, third one is the vernalization pathway, and fourth one is the gibberellic acid pathway. Clear? And in the photoperiodic pathway you can see that the CO protein, or this is produced from the CO gene. This is very important for the floral pathway integrators. Okay, And in case of autonomous pathway, it stops the FLC, and if the expression of FLC, that creates the floral pathway integrators. And this is the vernalization process. Here also the activity of the FLC is stopped, and in that way the floral pathway integrators activates. And the gibberellic acid pathway, here the gibberellin directly play important role for the floral pathway integrators, or for the for the induction of the floral pathway integrators. Clear? And if this integrators pathway that becomes activated, so the flowering process will be continued. This is the important thing of the flowering process. Another part also there, the role of the floral organ identity genes. So I will explain that topic in the next video. So you continue with my channel. Okay.